So we have only two different cells which are haploid, everything else is diploid. So the cells are sperm and egg, gamete, sex cells. That's it. Everything else what you were talking about in the animal body are the diploid cells, muscle cell, nerve cell, skin cell, the digestive cell of the stomach or lining of the stomach, epidermal of the bone cell. Is it a haploid or diploid? Is it sperm or egg? No, it's not. It's a bone cell. Bone cell will be diploid. So we are all diploid. So about zygote, is it egg or a sperm? No, it's actually a combination of egg and sperm together. So it is a diploid. So first two questions talking about the same thing. Fourth cell stage, how does the zygote divide by mitosis or meiosis? Let me kind of look over the, so fourth stage cell. So the meiosis, as you know, it creates the haploid cells. Haploid cells are only egg or sperm. Is it egg or sperm? No. Therefore, it have to be mitosis. Okay. Yes, I'm recording it right now. I think I'm recording. Yes, I am recording it. Yes. So in this particular case, mitosis will be the process of how the cells are being produced. 8 cells name the cell which creating the hollow ball. Whenever you're looking at this particular stage, it's a hollow because it has a cavity inside, the cavity known as a blastocell. The units of that known as a blastometer. What the name of the hollow ball of cells known as the blastula. And the blastocell, blastopore, blastometer. There is no blastopore on this one. Why not? Because pore is the opening. This is when the cells become tiny little bitty and they cannot hold on to each other and they'll start collapsing, getting inside of that particular cavity. And this is the picture showing you they are kind of moving into the blastocell. Is a matter of fact that early gastrula, as soon as they, so blastopore is that particular opening, and depends what that particular opening will lead to. We have two very different groups, protostomes and deuterostomes. In protostomes, that initial opening, the first opening will become a mouth. Protostomes, first mouth first. And then in this particular, in the deuterostomes, that opening will lead to the anus, that particular collapse continue, the tube goes, and will merge with another pole. And that secondary opening will become a mouth, deuterostomes, right? All right, blastomeres, haploid or diploid? Blastomeres, is it the egg or sperm? It's neither. If it's not egg or sperm, it's a blastomere. It has to be a diploid. In the model of gastrula, identify archeron, blastopore, germ layer, sector, endo, and mesoderm. So we're looking at this particular structure right here. Fairly well-developed gastrula. You can see that that particular collapse continues and that tube is growing, and eventually it will become what? It will become a digestive system. So it will grow and will meet on another side, creating that digestive tube, which all of us have. And yes, it's not exactly the tube. It has lots of convolutions, etc. But if you think about it, if you can straighten it out, it's a tube. You have a tube inside your organism. So that one is the outside, inside of the organism. Make sense? Anyway, looking at the embryonic layers. Embryonic layers, the blue one, known as the ecto. Outside layer. You see it's on the outside. The yellow one is inside, endoderm. And now we have the red ones, the pink ones coming in, creating kind of moving in between the inside and outside in meso. Meso means in between, middle. All right, archeron, basically the ancient gut is all that particular structure. Blastopore is the opening. Germ layers, we got it. Name the stage when the ball cell starts to fold inwards. Whenever it starts folding inwards, so we have a blastula start collapsing, that's a gastrula. In the given two models, morula and blastula, how could you decide which one without seeing the blastocell? Number of cells. If number of cells you're like counting here and you're counting 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, up to probably 16 is morula. Over 16, probably 32, 64, that's most likely a gastrula. You're just counting the numbers, counting the cells. And of course, on the lab practical, if you'll see that, it's most likely a morula. All the gastrula actually show the blastocell. So easy. Okay, how many germ layers do animal with bilateral symmetry have? Bilateral, us, we have 3. The only one which has the 2-layer diploblastic cnidarians and ctenophora, and they are generally radial symmetrical. Please do not get mixed up with the echinoderms. Echinoderms 5 radial symmetry, but the larvae is bilateral, free swimmers, so they belong to the bilateral. Yet they ended up with the radial symmetry kind of secondary. All right, know the different phyla covered in this lab and examples of animals in each phyla. Okay, number of germ layers, body cavity, present, absent, what kind of body cavity, true or false. Body symmetry, radial or bilateral. Digestive system, no digestive system, gastrovascular system, which means what? A sac. And then the complete digestive system, a tube. So phylum Porifera, know the numbers of members of the Porifera. We're talking about the sponge, the bass sponge, the coral, no, not coral, the calcer sponge. Know that these particular structures, like 6-ray stars, known as the spicules. This is a glass sponge. This is a colonial sponge. Here's the glass sponge spicules under the microscope. Quite nice, like a Mercedes, and I'm not advertising, but 3-ray stars. Mercedes does not pay me much. They don't pay me at all. And I don't have Mercedes in my driveway. I don't want. So anyway, in this particular case, name of distinguish sponges from different from the plants, because if you'll think about it, they look similar, kind of similar to the plants, sitting doing pretty much nothing. But the plants are photosynthetic, most of them. They can produce their own food. They are autotrophs. Remember that particular reaction of the photosynthesis? Yeah. So in this particular case, the sponges have to grab something. They filter feeding. What they are filtering out? They're filtering out living organisms, and then they will digest them. So and they'll kill them. They cannot photosynthesize. How many germ layers do those animals have? That's not a really correct answer, but let's say 2 for simplicity's sake. Be able to list characteristics of the phylum. So what exactly the characteristics of that phylum? First of all, no true tissues. No tissues. The cells can exchange. Like if we would be belonging to that particular group at some point of time, the cell from the skin decided, I don't want to be a skin no more. I want to become a muscle, and it will become a muscle. Or I will become a nerve, and then you'll have a nerve sticking out. I'm simplifying, but this is generally what they have. And then, of course, they don't have a true symmetry. They asymmetrical. They don't have a digestive cavity whatsoever. They have an intracellular. The digestion happens within the cells, a specialized cells, but it's the cells. They're not creating any kind of cavity. And I think, yeah, do they have a digestive system? No, they do not. How do they capture food? They filter whatever is captured. Well, they have amoeboid cells, which look like amoebas. They kind of moving around, surround the food, digest it, and then spread the nutrients to the neighboring cells. Pure intracellular digestion. Label the basic structure of the sponge, including the ostium, osculum, spongocell. So when we're looking at that particular creature, right here, So you see tiny little bitty openings. That's the ostium. One bunch of them ostia. Now that's a large opening. That's the osculum. Water gets from all over the sponge and then come out from that particular large hole, not in reverse. And I think there is a no the direction of water through the sponge. From all over the environment, larger surface area, larger the interaction. So more plankton can be get into the sponge, and then it comes out from the one opening. And the spongiocell, basically that particular dark cavity inside, if you look right here, that cavity right there inside, that's the spongiocell. All right, so we have that's the spongiocell. If the sponge looks like this, so here we have ostia, that's the osculum, and that particular cavity, but it's kind of hard to point it out, so I doubt that it will be in the lab practice. What makes up of endoskeleton of the sponge? Spicules. Spicules. Know what makes up the endoskeleton of bass sponge, Venus flower basket, or colonial sponge. So, bass sponge. Some silica dioxide and calcium carbonate spicules. And of course, the protein, demospongin or spongin. And then we are talking about Venus flower basket. Let's see this guy or girl. They asexual technically. So this one will be made predominantly out of silica dioxide. In the real world, well, in the real, in the face-to-face, I shouldn't say real. We have real lab right now, but in the face-to-face lab, whenever you see it, some of the students, well, it says do not touch, but can I touch it? You know, that particular desire when you say do not do something, like I want to do it. Yes, you go ahead, and if you touch that one, it will feel really, really kind of harsh or very weird because of the silica spicules. They kind of start poking your fingers. Not a very pleasant, not a very pleasant feeling. All right, and then be able to recognize glass spicules. Here we have this is not a glass spicules. This is the spicules made out of calcium carbonate. And here we're looking at the glass spicules. Already talked about those. All right, so yeah, spicules, spongin, boss. So here we have those ones. Now we're looking at the next group known as the cnidarians. And cnidarians, how many germ layers the cnidarians have? Two. They have ecto and endoderm, no mesoderm. Jellyfish, no muscles, or almost no muscles. Why not? No mesoderms. Most of the muscles made out of mesoderm. What is the symmetry of this group? Now they do have a symmetry, but it's a radial symmetry. Name the characteristics of this phylum. Gastrovascular cavity, radial symmetry, diploblast. Okay. Now know the difference between two body forms. The tentacles up, that's most likely a polyp. The tentacles down, that's most likely a medusa. So looking at this hydra, tentacles goes up. Is it a medusa or it's a polyp? It's a polyp. All right, so now a jellyfish will have a medusa predominant, and it's a mobile. The anemone is the polyp, usually a single one, and it's a sessile. Coral, usually a polyp, a colony of the polyps, and usually a sessile. Abelia, I'll show you the pictures of that. That's technically a polyp, a colony of them, have both body forms, and it's a sessile. I'll explain that momentarily. And the hydra, as you can see on the picture model, it's a polyp. It's a singular one. It's kind of mobile. It usually stands and waves, do nothing, but whenever there is nothing captured, it can do the seesaw thingy and move to the different spots. So technically it's kind of mobile, but not very fast. All right, what are the distinct characteristics of phylum? We just talk about that, name the characteristics. It kind of repeats itself. What the function of the nematocyst. You remember that one, the cnidocyte containing nematocyst. What the nematocysts do? Triggers, it shoots out some sort of toxin. What for? A, to disable the prey. Remember, they are predators. They are very successful predators, but they slow. How they can capture the fish? Kill it somehow. Fish, for example, and you probably seen the Nemo. There was a case when they were jumping on the jellyfish and the father Nemo and the Dory, right? And the Dory got stung and almost got eaten. Yes, very horrible part of the movie. Anyway, and what else? They slow. Slow creatures usually will be preyed on, so it's also a defense mechanism. If you ever been stung by the jellyfish, I've been not a pleasant thing. But in this particular case, why? Were it tried to eat me? No, I'm too big. But it was thinking. It didn't think. It just automatically I brushed against and I was like, here we go. Don't come near me. I'll kill you if I can. So this is the function. The hunting and the defense function. Abelia slide. I have a wonderful combo slide of Abelia somewhere. Oh, right here, right there. As you can see, colony of the polyps, and the colony of the polyps have two different parts. The feeding polyps, the tentacles, to capture and eat and distribute, digest, and then throw up. Whatever is not digested. And those ones contain tiny little bitty round things. That's the tiny little bitty medusas. They will literally shoot them out, and then the medusas will float, and then another medusa, and they will have the sexual reproduction, and then new abelia will start growing. Make sense? For the sexual reproduction. So feeding polyp does need tentacles to get to the prey. Why the polyp contain medusa bud? For the sexual reproduction. Hydra model. Yeah, it's all over the lab. So Hydra model right there. Be able to identify the following structures on the model. Mouth, right here, right in the beginning. The tentacles, the things sticking up. So it's a polyp. Gastrovascular cavity. After the food gets inside, it gets into a gastrovascular cavity. This is where most of the digestion will occur. So it's an extracellular digestion. In sponges, it was intracellular, predominant. Pretty much all the intracellular digestion. Here you have extracellular digestion, and yes, partially will be intracellular digestion. Even us have the intracellular digestion. White blood cells will capture the bacteria and destroy it. How digesting? Yummy bacteria. So anyway, gastrovascular cavity, cnidocyte. The whole thing is the cell. The nematocyst is that particular barb thing. Nerve net. Nerve net will be located between the ectoderm and endoderm on a larger picture. If you increase the size, let me see. You can see that particular yellow thing. That's a nerve net. They don't have the central brain. It's basically the cell connects to another cell, creating like a network of cells. The nerve cells, that is. But they come up with the nerve cells, which, by the way, video, which not all of you watched it, was talking about. So please, please, please watch the videos. They're helpful. Otherwise, I would not be assigning them. If it was just like, hey, watch a video 2 hours long. One of my sons in high school right now, and I'm like, why in the world are you watching a video? And he's like, well, it's a sign. And I'm like, are you learning anything? He's like, it's just a sign. Anyway, and I'm sure 10 minutes, 15 max. So anyway, looking more nerve net, we already did that. Now let me move lower. The asexual bud. Here we have the asexual bud. Eventually it will separate, will be a mini-me of the hydra, and the testes and ovaries right here, but you don't have to worry about that for the lab practical. Phylum Ctenophora. Ctenophora is very interesting, look like a medusa. See right there, a nice Nice creature right here. Again, with that particular dome, the tentacles. The tentacles lack of nidocytes. No nidocytes. They have a sticky substance, protein based. This is how they're capturing, but no nidocytes. You can grab it and it will not sting you because nothing, there is no stinger, so to speak. And also, they have something which known as the floating. I think I addressed that floating anus. They don't really have anus per se, but whenever the gastrovascular, and the yellow is the gastrovascular cavity filled up with all kinds of undigested part, there will be an opening random or somewhere on the random. As far as we understand, we don't know much about those creatures, how exactly it formed, but essentially there will be opening somewhere on the side of that where everything will be expelled. Can it throw up? Yes, it can. It can get rid of the excess of undigested material through that, but also it kind of moving toward the formation of the tube instead of just a sac. All right, now we're done with that particular one. Let me kind of pull out, don't say. Let me pull out the other, the lab number 12. Yes, that one. So lab number 12. Oops. Lab number 12. So flatworms, know the members of the phylum be able to recognize the preserved specimens. We're talking about those guys. That's the planarium. We're talking about those guys. That's the liver flukes. And we're talking about either these guys or that guy or those creatures. These all are the flatworms. What are the characteristics of the phylum? Flattened body, bilateral symmetry, triploblastic, no xylem, asalamate. Special characteristics. Now, sensory system. Comparatively to the Nidarians, now they have a front end. Front end will have sensors. Sensors will lead to the brain. Simple brain, yet it's a brain. It's a concentration of the nerve system in the front end. Ta-da, brain. Head. And of course, we're talking about auricles. It's not really ears. They don't hear as far as we know. But the eyes or eye spots, they don't really see. They can recognize the light and the darkness. And then they have a very nice gastrovascular system. Gastrovascular system, the black branched out things. They don't have a circulatory system, so the digestive system does that. It's still one end, a sac with the branches out. So this is the pharynx, so this is the mouth. This is how the food gets in, and this is how the waste comes out. Same thing on this particular model. You can see that the dark reddish maroonish color, that's the gastrovascular system, pharynx, the eye spots, and the auricles. So I think what else? What is special characteristics? Sensory system. So cephalization. They start having the head, be able to recognize the structures. Eye spot. I already showed you. Auricle, pharynx, gastrovascular cavity, and same thing on this one. The auricles, those kind of ear-looking thingies, and then the eye spots. That's kind of given the pharynx in the middle. It's a tube, it's a feeding tube, which also was showing in videos about the flatworms and the gastrovascular cavity. Do liver flukes live where? Do liver flukes live in the human body, in the liver? What are the other hosts? Do they live after they leave human? Mollusks and the fish, generally speaking, or mollusk and the other vertebrate, but mollusk and the fish. Know that liver flukes are hermaphroditic. Know the definition of hermaphroditic, being male and female or having male ovaries and testes at the same time. What is the advantage of this strategy? Self-fertilization. They can self-fertilize themselves to produce eggs, diploid organisms, next generation. What is the disadvantage? Self-fertilization is disadvantage. It's advantage and disadvantage at the same time. What does that mean? Whenever you self-fertilize, or whenever they self-fertilize, they drastically lacking the genetic variants. And sexual reproduction, main idea is to increase the genetic variants. Where do you, okay, oh, how the humans acquire liver fluke? They eat in the intermediate host, not fully cooked. This is how generally we're getting. So those eggs have not been, well, those larvae have not been cooked. As a result, we are eating them, and they are going their own way. So, okay, I recognize the following structures on the liver fluke, ovaries, testis, uterus, gastrovascular cavity. Now, when you're looking at the model now, so the gastrovascular cavity, that particular red lines, they're going along the body, supplying the body with the nutrients. Where are they getting the nutrients? Straight from the blood, essentially. The brownish structures right here, all of that is essentially the ovaries, and the blue ones would be testes. So the sperm producing goes into the ovary and will fertilize the eggs. So they will produce tons and tons of eggs. What is the advantage of this strategy? Okay. What is the insect vector which transmits tapeworm to dogs? I think in your lab it's talking about the fleas, right? They chewing down on the flea and it gets into the digestive system. How a person can acquire the tapeworm? Well, chew on the same flea. No, no, not really. We generally don't do that. But technically, yes, if you accidentally inhale, then it will end up in the digestive system. There we go. But quite often we getting the tapeworms from either uncooked or poorly cooked meat, or we might get from our pets. Not washing, you pet the infected dog, not washing your hands, taking the food. Here we go. So yes, if you're petting an unknown dog, it's a really good idea to sanitize. Nowadays, we're all about sanitizing our hands. Yeah. So I even seen the people who doing the lectures in a mask. I hope you're okay with me not doing that. Anyway, the tapeworm body highly specialized. Do they have a digestive system? No, they don't have the digestive system. They live already in a digestive system of the host. So they're taking all of the dissolved nutrients from there. Be able to recognize the hooks and the suckers. So here we go. This is the scolex, the head, so to speak. Here we have a bunch of hooks, and here we have, oops. Okay, and here we have the suckers right here. They are sucking to the intestinal wall. Why? Not to be removed, because intestinal wall is slippery, so they can slip out, and then the movement of the digestive tract can get them out. So this is why they need to attach, so they will not slip out. Okay, scolex, we did that. What function? Attachment to the intestinal wall. Mature troglodyte will have ovaries and testes, and the gravid troglodyte will have the zygote. And we're talking about this particular model. We have the immature, now mature troglodyte. You can see the ovaries, the yellow part, and the testes, the pink part or red part. The sperm comes out from this opening, goes straight into the oviduct, into the ovaries, fertilizing those particular eggs. And now look at the gravid troglodyte containing tons of eggs and pretty much no longer have any testes, no need for them. The sperm has been used to produce hundreds of thousands of eggs. Now that particular segment can separate from the body of the worm, go into the feces, and be done with it. All right. Well, and get into the water, and the whole cycle continues. What type of the troglodyte can break a body through the body? That would be a gravid troglodyte. A phylum of Annelida. So next Don't take. Next, echinoderms. No, I need the other 13. There we go. Annelida, yeah. No, 12. I need 12. 15, 13, 14. I didn't open 12. Okay, let me open 12 really quick. All right, so lab number 12 and lab number 12 talking about those guys. All right, so we are talking. Oh, no, no, it was here. So we are talking about, yes, I kind of stuck on the. So here we're looking at the Annelida, earthworm and such. What distinct characteristics of this phylum? Segmented body, first. They still bilateral, they still have head, so, but segmented body, closed circulatory system, and now they have a complete digestive tract from the mouth, intestinal crop, etc., and then anus. It's a tube. It's no longer a sac. What is the nature of xylem in this phylum? They solemate, meaning that the body cavity located between the mesodermal, within the mesodermal layer. On a model of earthworm, identify mouth, pharynx, aortic arches, digestive, well, crop gizzard part of the digestive system, and seminal vesicles, seminal receptacles. You don't have to worry about clitellum and segments. Yeah, you probably should. So let's kind of take a look on that picture. Oops, too much. So on that picture right here, so where are we looking at? This particular part, kind of like a band on the front end of the earthworm, that would be the clitellum segments, those repeating units. Now, mouth, of course, is right here. This is the green tube with the digestive tract. Now, this is the aorta, and then we're looking at the aortic arches. Aortic arches is essentially hearts. They are increased, they have a thick walls, and they pulsating to move that particular blood. So that's pretty much all you need to know. Be able to identify tube worm, leech, and marine worm. So here we're looking at the marine worm, right? Clam worm, marine worm. Yeah, looking kind of weird, but they essentially harmless to us at least. Here we have a tube worms, and the tube worms will create those tubes where they're living inside, or could be on the surface of the on surface of any kind of well, shells or piers or anything. Can be done with the hull of the ships also. And then we're looking at those ones. So let me decrease the magnification. So here we're looking at the earthworm, a leech, and the scale worm, which is a marine polyhead. So again, we're looking at the leech right here, and then the marine worms and the earthworm. All right, mollusks. Mollusks will go over the bivalves. On the snail, we don't have the preserved open or model of the snail, so you don't have to worry about visceral mass and mantle. Be able to recognize the head, the foot, and the shell, right? Be able to identify the lightning whelk, the official state of Texas mollusk, or mollusks of the state of Texas, this guy. And then we're talking about the slug and the murex, relatively easy. Now Nautilus and the octopus. So here we're looking at the octopus, another octopus, 1, 2, 3, 4, 5, 6, 7, 8 arms slash tentacles. And this is, of course, would be the squid. And you can see that two of them are much longer. Those ones are known as the tentacles. Those, the shorter ones, known as the arms. And the Nautilus, very characteristic shell with the lines, chambers inside, and here's the preserved guy. Oops. So the preserved Nautilus is right here. You can see the same shell with the mollusk in there, and that's the fossilized nautiloids. We can see the round and straight shell. All right, now let's see following structures on the model. So here we have the model right there, and they all kind of marked for you. But let's kind of go into an unmarked picture of that, and let's see. You will need to identify the mantle, that particular kind of covering of the all of the internal or visceral mass, internal organs. So that part is the mantle. Then we're talking about the food, right? Visceral mass is all of the internal organs. Abductor muscles, the one which is closing the shell, and then whenever they relax, the shell springs open. Then the gills, that brown things with the lines, this is where water comes in and bringing the oxygenated oxygen will be extracted, carbon dioxide could be released. And the heart is right here. That's the heart. And that's the xylem, that's the body cavity. The heart located within the xylem. So they have a true xylem, and that's, of course, its digestive system. Okay. Identify oyster and a scallop. Oyster and a scallop. Here we go. Oyster, scallop, spiny oyster, abalones, mussels, all of them pretty tasty if you're in the seafood. Some people don't like it. Well, I do. Be able to identify on a squid. Now let's take a look on the squid, this guy right here. And it could be a little bit tricky. Some of the structures kind of hard to see, but look, that one is definitely longer than this one, right? So this is the arms, that's the tentacle, as a matter of fact, two of them. The suckers on top of the tentacles as well as the arms. And then we have the siphon right here. That would be gills. So the water goes through the gills and then will be, actually it will come from the other side, goes through the gills and then will be sent out. Now all of that, you see, that particular covering, it's being cut out to show the visceral mass. That's the mantle. Technically it stays closed, and that's why whenever you have calamari, the rings, what is it? It's basically you eating mantle. The internal part has been removed, the mantle has been cut into a nice rings. Here we go, fried, and you enjoy them. So that's basically it on the, oh man, I need to go faster. That's basically it on the mollusks, right? Yes. Okay. Phylum Nematoda, roundworms. Again, some of the nasties. Yes, here we go. Again, whenever you're looking at the specimen, make sure to know which one is a male, which one is a female. Males usually smaller comparatively to the females, and all of them will have hooks on the tail. Trichinella, be able to recognize the dark brown color, that's the muscles. The whole round thing is the cyst. The thing inside of the cyst is the larvae. Why do we have to cook meat to 145 degrees Fahrenheit at least, usually 160? But in this particular case, to cook those creatures, you will not cook them out, you just cook them. And if you're eating infected part, you will still eat them, but they will be cooked, so they will not develop into a worm inside of you. Uterus, vagina, and ovary, don't worry about that. It's really hard to see, so I will omit that. Just believe me, if it will be on the lab practical, yeah, those ones. If it will be on the lab practical, I'll just give you the points for that. Be able to recognize the heartworm. You see those particular stringy things? That's the heart. Well, that's the heart, and that's the worm inside of the heart. So don't tell me it's a dog heart. No, I'm talking about if the question is, we'll say, name the organism, and it will be pointing at the worm, then it's most likely I'm not talking about the dog. I'm talking about the actual worm. Arthropods. Yeah, what vector transmits the heartworm? Mosquitoes. Be able to identify. Okay, we're done with that. Don't save. Now, grasshopper. on a crayfish or crawfish, whichever you prefer. On the crawfish, be able to recognize gills, those particular feathery structures right here. You can see them much clearer right there, and that's the gills right there. Now, telson. Telson is kind of hard to see, especially on the model, but on this one, I think, let's see, on this guy. On this guy, you can see the telson is the central part of the tail, so to speak. On the very last end, the central part. On this guy, you can clearly see that one would be the telson. Cephalothorax. Cephalothorax is basically on this one, you can clearly see it's a head and the chest together, and this is the abdomen. Then what else we need to see? Gills, telson, cephalothorax, abdomen, walking legs, the walking legs right there. Now swimmerets. Swimmerets will be kind of underneath of the abdomen. You will see those tiny little bitty looking structures, but much smaller. They send the water toward the gills. Swimmerets, antennas versus antennulus. If you look at this guy, the large ones are antennas, the large one right here. The smaller one, antennulus. Rostrum, the thing which is sticking out in front of the eyes and the compound eyes. So here's the rostrum, here's the rostrum, compound eyes. Pincers. Well, I kind of understand that everybody knows pincers, compound eyes, done. Following the structures on the grasshopper outsides. Spiracles. Spiracles are tiny little bitty opening on each of the segments. Here we go. This is how the air gets in. Then we need to look at the head, compound eye, antennas, the wings. Then we need to look abdominal, thorax, compound eye, spiracles, ovipositor. The very end of the abdomen has that structure. This is how the eggs come out from the ovaries and will be laid down somewhere in the grass. So the little one will be hatching out and start jumping on the grass right away. Knowing the structure on a grasshopper, internal view. So what we're looking at, a mouth, well, head, mouth, the nerve system, the brain, now the ventral nerve channel right there, nerve cord. Then we're looking at the digestive system and those kind of finger-like projections. This is a gastric ceca. Gastric ceca, that would be the ovaries with the eggs. So what do we need to see? Mouse, brain, ovary, gastric ceca, ventral nerve cord, heart. Here we're looking at the orange blood vessel and we're looking at the thickening of the blood vessel walls. That's the pulsating walls and that's the heart. Identifying metamorphic stages of the butterfly. So in the butterfly, what do we have? We have the egg, larvae, pupa or chrysalis, and the adult. On the grasshopper, we'll see the egg, nymphs. If you can call them larvae, it's K-ish, but better I'll call them nymphs. And then the adult grasshopper. Be identified the stages, how many legs? 3 pairs. Define exodus. Exodus is the process of shedding the old exoskeleton and putting, to grow. Characteristics of the phylum of Arthropoda: jointed leg, exoskeleton, complex nervous system, compound eyes, all that. No insect metamorphosis, talk about that, giving the representative of the insects. We're talking about those ones, the Lepidoptera, the butterflies, the dragonflies, Adonata, the Arthroptera, grasshoppers, and the crickets. Then the Diptera would be the flies, mosquitoes, and such, horseflies, Dermoptera, don't have to worry about that. Then we have the Hymenoptera, that's my favorite one, wink wink. So Hymenoptera, that's the bees, bumblebees, wasps, ants, and such. And then we're talking about coleoptera, the largest one, so the beetles, and then we have a hemiptera, that's the true bug, like assassin bugs, etc., and homoptera cicadas. De-identify structures on a grasshopper, we don't have those ones. Cephalothorax, now we're going to the centipede. Centipedes, centi means 100, so centipede, one segment will have one pair of legs, while the one segment of millipedes have two pairs of legs. Identify following structures on a horseshoe crab. Horseshoe crab, the cephalothorax, technically prosoma, the abdomen, opistosoma, telson, that's the telson, not a stinger, telson. Now the eyes and the book gills right here, and that's the walking leg. So now if you look at the scorpion, oh, not scorpion, the spider, right? I think with spider. No, we need to do this scorpion. Scorpion, the preserved specimen, we have the pincers, the eyes, cephalothorax, walking legs, the stinger. Now on the spider, we're looking at the walking legs, and those ones kind of tiny walking legs. They call pedipalps, and then the fangs. So 4 pairs of walking legs. De-identified, don't worry about that. Deadlift mantis right here. So the head, the thorax, the walking legs, the grasping leg, and then the wings and the abdomen. Bilateral symmetry. And we are done with that one. Echinodermata. On a C-star model, be able to recognize, let's kind of take a look. Digestive glands. So let's see, digestive glands, the green feathery stuff, blue or bubbles, that's the ampullus, which will lead to the tube feet. Also had that in the video. Madreporite, sieve plate right here. Mouth will be on another side. Here we have the mouth. This is the rows of the tube feet. Umbilical ridge. Ridge is right here. That part in the middle is right here. This is the ridge. And then what else? A ring canal. You don't really see ring canal here, but you can see ring canal here. So we have a madreporite or sieve plate, swan canal leading the water into the ring canal. Ring canal will split it into a radial canals, which will fill up ampullus and the tube feet. Water vascular system. And then what do we have? Sea urchin. So here we have the examples. Sea urchin, pencil sea urchin, sand dollars. So sea star, sea urchin, sand dollars, galaturia, which is a sea cucumber, brittle star. And here we have brittle stars. Here we have live galaturia and the preserved specimen. Entry point of water will be a sieve plate, deuterostome. This the mouth developed from the secondary opening in the gastrula. Do members of phylum have internal or external skeleton? Internal skeleton. On lab 15 we covered that pretty much, but just in case, knowing be able to recognize the preserved animals. So we're talking about the something like a puppy, a bat, kitten, chicken. There's snake, rattlesnake, tortoise, right? And then we looked at the frog, the salamander, different types of fish, and that's a bony fish, the a seahorse, and then the perch, and then of course the skates and the sharks. Oh, that one I didn't talk about that. This is the eggshells of the shark, similar we see in the skates, and they call it quite often, common name is the mermaid purses. So, distinct characteristics, we went all over that. The sharks and scales, the teeth made out of denticles. Now the following structures: dorsal, pectoral, pelvic, gill slits, ventral, mouth, spiracles. Make sure you know those. Following structures: dorsal, pectoral, pelvic, anal, terminal, mouth, operculum, the covering, and the swimming bladder. Sternum. Sternum is the keel and the function attaching of the flying muscles. And then we have members. You can basically fill it up by yourself. If you're not really sure, send me the email, kind of check on that, and we're pretty much done today. So once again, thank you very much. See you next week for the lab practical. Yay! And you have till Sunday night to um, to do the test. Also, let me kind of stop sharing and stop recording.